Welcome everybody to this week's edition of the Extracellular Vesicle Club. So it's my honor today to introduce you to Setty Magaña. She is an attending pediatric physician at Nationwide Children's Hospital, and she's recently appointed as assistant professor at The Ohio State University. So Dr. Magaña, thanks so much for joining us today, and I would like to hand the screen sharing over to you to give your presentation.
All right. So it is truly a pleasure to be able to speak to you. I am relatively new to the EV field and have really enjoyed getting to know some of you, and hopefully through this presentation, I can get to know more of you. So let me see here if I can go to my next slide. So I have no disclosures. So I'll be giving an overview of pediatric neuroimmune diseases. And then for reasons that will hopefully become apparent, I'll be talking about adipose tissue as an active immunological organ, and then give an overview of the EV omics pipeline that we're using to study MS, and then share some works in progress. So I want to kind of land on this slide just to give some background and some context. So I am a pediatric neurologist. I did my PhD at Mayo Clinic in adult MS and also a related disorder called neuromyelitis optica. But it wasn't until my residency when I worked with a adult brain surgeon who was doing work on EVs in GBM that I learned about EVs and non-coding RNAs, working on a pediatric brain tumor project, which is currently under review. But that's really how I got interested in EVs and really wanted to move my research focus to be EV centric. And I saw a lot of potential application from what I learned in the pediatric brain tumor space to other CNS inflammatory diseases. So I am going to focus on multiple sclerosis today, but there are a spectrum of diseases that of patients that I see. Today, though, I'll focus on MS. So I wanted to start really giving you a clinical context with a patient that I saw in our hospital a couple of weeks ago. So this was a 12-year-old female who presented with left hand numbness and weakness. And so she was noted to have some pain and numbness in her left index finger upon awakening the day prior to her evaluation. And due to some new onset weakness, her mother brought her to the emergency department where they called a stroke code. And so on review of systems, she had some associated postural lightheadedness. She did not have any vision or bowel or bladder symptoms, which are important to exclude. She was noted to have a febrile illness 2 weeks prior to this. She was COVID negative, but several family members were COVID positive. Otherwise, her past medical history was unremarkable. Her family history was notable for a paternal aunt with multiple sclerosis. And then on physical exam, she was noted to have mild left-sided upper and lower extremity weakness, as well as some sensory symptoms along her C7 dermatome. So this prompted brain and spine imaging, as well as a spinal tap. And so these are her brain images, just a few of them to highlight some of the findings. So for those who are not used to looking at brain MRIs, on the top here, we have axial brain images where the patient, the patient is on their back and it's as if you're standing at the foot of the bed looking up. So this is the right, this is the left, the front and the back. These are the CSF-filled ventricles. This is a T2 weighted sequence where the white matter appears darker and the cortical gray matter is a bit lighter. And so here we can see, hopefully they project some periventricular lesions here, as well as a subcortical lesion here. On the flare sequence, it's similar to the T2, but it takes away the bright CSF signal so you can see the lesions a little bit more clearly. And then this is a T1 contrast enhancing study where it's faint, but there is some mild enhancement of the lesion here. And so that indicates active inflammation with blood-brain barrier breakdown. This is another view. This is a coronal view, like you're cutting right down the head here. Again, right and left, periventricular lesion here and here. And so we also obtained her CSF analysis. This is just an example. This isn't her gel, but she did have some oligoclonal bands that were unique to the CSF, not present in the serum. And then this is her spine imaging, so front and back. This is, again, a T2 image where the CSF is bright. And you can see this lesion here about the level of C4. And then this also had some enhancement. Let's see here. So at this point, she does meet the clinical criteria for MS. I will note that there is no specific marker for MS. So in fact, up to 30% of patients who see us in a MS specialty clinic are misdiagnosed as having MS. So the diagnostic criteria require that there is dissemination in space, which can be demonstrated by lesions in at least 2 of these locations. So she did have periventricular, she had spinal cord lesion, she had subcortical lesion, and then dissemination in time, which can be demonstrated by at least 1 enhancing lesion, which she had, and at least 2 oligoclonal bands in the CSF. So she received a first-line treatment, which is IV methylprednisolone, steroids for 5 days, and then she was discharged on a steroid taper, as well as instructed to follow up with physical therapy and also outpatient MS evaluation to discuss lifelong maintenance therapy. So this is just to show the clinical course and phenotype of MS. So we know that there's a pre-symptomatic phase where there are lesions that are accumulating, but they don't meet a clinical threshold. So they don't have any symptoms. Then patients will present with their first attack. At this point, you don't know whether it's an isolated attack or, they will convert to relapsing remitting MS, which is the most common kind, where patients will have discrete relapses. They may return to full recovery, or that recovery lessens as time progresses. And then these patients can convert to what we call secondary progressive MS. A minority of patients will start out with a progressive course. So from a clinical standpoint, some of the questions that we have to have with patients and their families, when was her true disease onset? So she had various lesions. The brain lesions did not correlate with her clinical symptoms. So when did she first start to have these lesions? Particularly for pediatric patients, will this affect the overall brain growth and her cognitive function? There is evidence to suggest that pediatric patients with MS do have decreased brain growth compared to healthy peers. Will she have frequent early relapses? At this point, we have no way to prognosticate or predict the course that patients will have. And will she achieve complete recovery in between relapses? And importantly, what is the best maintenance therapy for her? So just a brief background on multiple sclerosis. So it's the most common cause of non-traumatic chronic CNS dysfunction in young adults. 85% of patients will experience reversible episodes of neurologic deficits or relapses that last days to weeks. MS is a heterogeneous multifactorial disease. It has no known cause or cure. It is known to exhibit a complex interplay of genetic and environmental risk factors and substantial immune-mediated neurodegeneration. And this is work by my PhD advisor, Claudia Lucanetti, showing the pathological hallmarks of the disease. So there are focal demyelinating plaques that can occur anywhere along the neuraxis. So that includes optic nerve, spinal cord, and brain. There's evidence of post-capillary venule blood-brain barrier breakdown with inflammatory infiltrates, including CD8 T cells, CD20 B cells, some CD4 T cells. These are active astrogliosis. And then you can see lipid-laden macrophages with relative preservation of axons. And so in terms of the immunopathogenesis, here you have the periphery and the CNS compartment. This was historically considered to be a T cell mediated disease, but now we know that there is bidirectional crosstalk between the peripheral immune cells and the CNS resident microglia and astrocytes. All our current therapies primarily are targeting the peripheral immune response and are very poor at addressing the compartmentalized CNS inflammation. The primary effector cells are Th17 cells and Th1 cells, but there is evidence that there are aberrant Treg, CD8 T cells, and B cells that result in a pro-inflammatory milieu with peripheral cells being recruited to the CNS, causing infiltration in this CNS compartmentalized space. And so for pediatric MS in particular, most patients do manifest symptoms between 20 to 40 years of age. However, up to 10% of patients will have childhood or adolescent onset of their symptoms. Prior to puberty, MS occurs equally in males and females. After puberty, there's a 3-to-1 female predilection. And it's known that pediatric MS patients exhibit distinct phenotypes and trajectory compared to their adult MS patients. So they have earlier and higher relapse rates, so almost 3 times that of adults. They have a higher lesion burden. They have early cognitive impairment, as well as brain atrophy. And as I mentioned before, recent imaging study suggests a subclinical onset of MNs that commences very early in childhood and negatively impacts brain maturational growth. And so because of this, pediatric MS offers a unique window into disease pathogenesis that is temporally associated with disease onset. So as I mentioned, MS is characterized by a complex interplay of environmental and genetic factors, with the largest genetic contributor being MHC on chromosome 6. Some of the environmental risk factors include low vitamin D, viral infections, including EBV, passive smoking, and obesity. And epigenomics have come into recent focus in MS pathogenesis, primarily looking at methylation status in immune cells. And epigenetic processes, including microRNA-mediated gene silencing, serve as mechanisms through which gene expression is reversibly modified. So this can be influenced by heritable, environmental, and biological factors, and helps to bridge the gap between genetic and environmental factors. So my interest is really at elucidating the mechanisms by which obesity increases the risk in MS. So childhood obesity has tripled over the last 30 years, paralleling increased prevalence of MS. And it's been shown that childhood and adolescent obesity is an independent risk factor for developing MS. Elevated BMI in genetically susceptible individuals is associated with a 41% increased risk of MS, with higher BMI around menarche being associated with earlier age of MS onset, as well as higher neurologic deficit in women, but not men. So the cellular and molecular underpinnings of adiposity-associated MS risk remain to be defined. However, adipose-associated cytokines, adipokines, and chronic low-grade metabolic inflammation have been implicated in pediatric MS, adult MS, and the animal model used to study MS called EAE. So now I want to turn our attention to what I have found to be a very fascinating area of research, really looking at adipose tissue as an active immunological organ. So adipose tissue has long been regarded as silent and a passive organ dedicated solely to energy storage and release. However, it's increasingly recognized as an active endocrine organ able to exert local and systemic homeostatic alterations. White adipose tissue in particular is composed of adipocytes, pre-adipocytes, fibroblasts, endothelial cells, and immune cells. And obesity results in adipocyte alterations in lipid and fat volume. This changes essentially the adipose tissue resident immune cell and circulating immune cell phenotypes that results in a pro-inflammatory milieu. And interestingly, obese individuals have 50% of their adipose immune cells comprised of macrophages versus lean individuals, where that's only 10%. And even in healthy individuals, 30% of circulating IL-6 originates from adipose tissue. So these are some, just highlighting some of the articles I've read that were fascinating to me. So this particular study observed adipose tissue T cells dynamically interacting with adipose tissue macrophages, demonstrating that MHC2 in these macrophages is required for obesity-induced insulin resistance, as well as to generate effector memory T cells, and that ablation of CD11C positive macrophages in obesity attenuated the conventional T cell proliferation, identifying potential therapeutic targets for obesity-associated meta-inflammation. This study looked at high-fat diet versus control diet-fed mice that were infected with lymphocytic choriomeningitis virus. And then they looked at the effects of white adipose tissue and obesity on T cell responses to infection. So they were able to show that the virus replicated in the white adipose tissue. Virus-specific T cells cleared the virus and then persisted as memory T cells. These memory T cells then underwent physiologic changes that were caused by the obesity, making them phenotypically distinct from T cells in other lymphoid organs as well as non-lymphoid organs. And then interestingly, upon re-challenge with a variant of the virus, the obese mice exhibited unusual forms of T cell-mediated pathogenesis. See here. And then this study, just a reminder for me to highlight that there appears to be a unique population of regulatory T cells that seem to be aberrant in obese versus lean conditions, which I will come back to later. This study, just a couple more. So this described these omental milky spots that are fat-associated clusters of leukocytes that may function as secondary lymphoid organs, containing macrophages, specialized B1 cells that have unique repertoires of antigen receptors, particularly with regards to identifying targeting bacteria. And so unlike conventional lymphoid organs, these omental milky spots develop in the absence of lymphoid inducer cells, but do require CXCL13. They collect surrounding antigen and support T-cell dependent B-cell responses. And in human studies, there's been evidence that human omental adipose tissue from obese patients undergoing bariatric surgery have these milky spots enriched for CD206 positive macrophages in direct contact with HLA-DR negative CD4 T cells. And so this got me thinking about just the potential role of adipose tissue in autoimmune disease. And there's some evidence to suggest that there's secretion of autoimmune antibodies in human subcutaneous adipose tissue. And then this interesting study linking the gut microbiome with white adipose tissue in obesity being regulated by microRNAs. And so adipose tissue is now considered a bona fide immune organ at the crossroad between metabolism and immunity. It contains conventional as well as unconventional immune cells. In one study that looked at 1,300 gene transcripts in the white adipose tissue, up to 30% encode inflammation and macrophage specific genes. And as I alluded to, some of the immune cells that reside in the adipose tissue are phenotypically distinct from lymphoid residing immune cells. For example, Tregs that rely on lipolysis rather than glycolysis. So this got me interested in looking at adipocyte-derived EVs in disease. And there are several studies out there that have looked at adipocyte-derived EVs in human circulation and their role in immune responses. Most of the work I found out there was in animal models. And so now with that context and background, I'll just turn to some of the work at NIH that I did and hope to do now at Children's and OSU. And so working with Jennifer Jones and Josh Welch, we developed an EV omics pipeline for studying EVs in CNS inflammatory diseases. Let's see here. Okay, so at NIH, all the patients that we see are enrolled in a protocol. So there's very detailed data collection as well as biofluid collection. So we would collect serum, CSF, urine, and saliva from patients, and then have all their clinical data as well as serological and imaging. And then we developed this pipeline for isolation and characterization of EVs using primarily the QEV columns, Spectrodyne for size and concentration, EM for characterization, as well as protein cargo analysis, and various RNA and multiplex and flow cytometry characterization. So most of the time that I was at NIH was invested in optimizing our EV isolation and characterization methods. The lab I was in did not have an active EV research program. And so we were just essentially trying to optimize these methods for human CSF and serum. So this is just an example of our QEV fractions from HTLV1 infected CSF and MS CSF, just showing that we had a lot of small EVs in our samples. We also had the luxury of having healthy volunteer CSF paired with serum, and just to highlight here that we have heterogeneous populations of EVs in the serum versus the CSF. So for our nanoparticle analysis, we use Spectrodyne. So for those of you who may not be familiar, this allows for single particle detection by these pre-calibrated disposable cartridges that have on-chip engineering with these filters. This is highly reproducible, and importantly for pediatric samples only requires 3 microliters of analyte. So you have your single particles here going through the nanoconstrictor, which generates a change in the voltage, which then allows you to determine the size, and then the flow rate allows you to determine the concentration. So when you compare NTA versus the gold standard EM and Spectrodyne, there's nice agreement for large particles. But as we noted, we have a lot of small EVs in our samples. So the Spectrodyne is what we decided to use. And this allows for better resolution of these smaller vesicles. So I'll just briefly go over some of the works in progress. As I mentioned, this is ongoing work. And so the first project was looking at bulk EV and microRNA repertoires in peds and adult MS. And so using this pipeline, we had pediatric patients from my collaborators at Children's National Medical Center in DC. We had 8 active pediatric MS patients, 6 stable. And then because of the interest with obesity, we had 10 bariatric pediatric patient samples from our adult cohort at NIH. Again, 5 active MS, 5 stable MS, and 5 adult healthy volunteers. There were 2 independent reviewers at each site doing the data collection blinded to the EV experiments. Because of the high volume of samples, we elected to do ultracentrifugation instead of the QEV. And this is just the early pilot data that I will be showing you. So this is the workflow. So we had 500 microliters of serum from both the pediatric and the adult cohorts. And then we had 2 tubes that we centrifuged to pellet the EVs. One was then resuspended directly in the Kysol, and then another tube was resuspended in PBS for the remainder of the characterization. At this stage, what we wanted to do was a pilot study using the adult samples, since the pediatric samples are very precious and hard to come by. This was the first time that we had done any RNA work in our hands in this setting. So we wanted to just make sure we could actually get some of the RNA sequencing here. So this is just the raw, it has not been analyzed yet, just to show for some of the pediatric samples, the concentration ranges here from the Spectrodyne. So anywhere from 1.84 e to the 10 to 2.82 e to the 10, not taking into account dilution factors yet. This is some of our EM results. Again, there was a lot of small vesicles in addition to other co-isolates from our preparation. But I'm very reassured that we were able to get some interesting results just even with the pilot adult study here. So initially the goal was to do deep RNA sequencing, but we had a lot of difficulty with the library prep for some reason. So we ended up using these Affymetrix microRNA plates. And so this is just the PCA plot showing no significant outliers. The MS stable is in blue. We had a progressive MS patient thrown in there that wasn't initially intentional, but interesting. And then the MS active in red and controls in green. So this chip has all species on it. So there were 638 differentially expressed genes, 79 of which were human. for the microRNAs, 31 upregulated, 48 downregulated. This is just the scatter plot showing control versus active MS, and then the volcano plot with fold change and p-value. So this is the heat map here. And what was nice to see, at least, was that the MS active clustered together. The progressive was separate from the control, and then the stable here in blue was more similar to control than to active MS. These are some of the top 10 upregulated microRNAs and some of the top 10 downregulated microRNAs. Again, this is just data I got last week, so I haven't had time to really go through it, but now we feel comfortable proceeding with our pediatric samples. So lastly here for works in progress, I was fortunate enough to get a grant to do this study. And so again, the central hypothesis is that obesity contributes to MS risk and clinical severity through the release of adipose derived EVs into the peripheral circulation, and that these EVs deliver pro-inflammatory adipokines and candidate microRNAs that drive neuroinflammation by modulating the peripheral and CNS resident immune cells. So for the first aim, we are going to isolate adipocyte-derived EVs from pediatric MS serum and characterize their cargo, particularly candidate microRNAs that have been implicated in MS and obesity. And then for the second aim, I'll be working with a pediatric bariatric surgeon who is going to provide adipose tissue from those patients. And then we will isolate EVs from those samples, and then incubate with PBMCs, lymphoid and myeloid cell lines and microglia, and characterize the immune response. And so future directions. So again, I've kind of highlighted this EV-centric research approach, and I think there are a lot of applications in the context of pediatric CNS inflammatory diseases with biomarker discovery. I didn't even talk about ADEM here, but is there a way to identify patients who will have a monophasic single attack versus those who will have recurrent or multiphasic ADEM? There's a spectrum of diseases here within demyelinating diseases. So there are some patients who present with what we call radiologically isolated syndrome. So they'll have a brain MRI and you'll see lesions there, but they haven't had a clinical attack yet. So how do we know if they're going to convert to CIS when they actually have their clinically isolated syndrome versus those who have an attack, and will they have relapses? So there's a lot yet to be done in terms of biomarker discovery, prognostication and therapy response, and then engineering EVs to deliver therapeutic payloads to the CNS, which, as I mentioned, our current therapies target the peripheral immune response, but not the compartmentalized CNS inflammatory response. And so this is just the overview of my research program. So I'm interested in studying the age span of multifactorial pediatric onset chronic neurological disorders by exploring the role of EVs and non-coding RNAs in epigenetic host dynamics using MS as the model disease. As I mentioned, there's a preclinical phase, and there's a lot of new evidence to suggest the role of EVs in pregnancy disorders with maternal fetal crosstalk. And so there are some prenatal and postnatal risk factors that have been identified for future development of MS, but the mechanisms are unclear. And really just the whole age span of the disease confers different risk, and the mechanisms underlying those risks are not well understood. And so as a pediatric neurologist, I'm also board certified in adult neurology. So my clinical practice will be to see pediatric and adult MS patients to better understand the age span of the disease, with the hope that we can identify and better characterize genetic and environmental factors which may be modifiable, better elucidate the pathomechanisms of disease at earliest onset. And then an area of great interest is modeling these processes using human-derived 3D model systems with the hope of developing precision nanotherapeutics. And so this is just a one slide from my collaborator at OSU, Alex Gardell, who was recruited to OSU to develop a biofabrication program. And so he has done some very exciting, nice work modeling blood-brain barrier and brain interfaces using human-derived cells with these defined brain mimetic platforms. And then also allowing study of the cerebrovascular unit. This has all been done in the context of brain tumors, but again, we could use this to assess drug or EV transport and bioavailability to the brain. So that's an active area of interest for us. And then lastly, so we have an integrative omics approach to MS. I've spent a lot of time talking about EVs, and I'll close by introducing this concept of the exposome for those who have not heard of it. So the human exposome was first introduced in 2005 to encompass the totality of human environmental, i.e. non-genetic exposures from conception onward. So this includes metabolomics, proteomics, transcriptomics, epigenomics, and it conceptually integrates overlapping and non-overlapping domains of non-genetic factors. So general external domains include social, societal, climate factors. Specific external domains could include lifestyle factors, tobacco, occupation. Internal factors can include the microbiome, metabolism, inflammation. So this is really uniquely poised to explore the role of environmental factors acting in synergy on multifactorial and chronic diseases. And the early life exposome allows the investigation of prenatal and postnatal factors. So I'll just refer to this publication that looked at the early life exposome in relation to pediatric blood pressure. And the neurological research community has shown some interest in the exposome in the context of Alzheimer's disease. So these authors proposed the AD exposome to comprehensively assess the modifiable environmental factors relevant to the genetic underpinnings of cognitive aging and Alzheimer's disease. So when I was talking to the exposome researchers at Columbia, and sharing my interest in EVs, then they were asking me, well, could there be an EV exposome, which I will leave for future discussion. But in conclusion, the role of EV and EV bioactive cargo is only beginning to be elucidated and offers a fascinating research field ripe for investigative inquiry, particularly at the interface of the developing immune and nervous systems. Elucidating the non-coding RNA cellular interactomes is likely ideally suited for multifactorial pathomechanisms at play in complex diseases. And the elucidation of EV-mediated epigenetic modification of host dynamics in MS pathogenesis has the potential to usher in a new era of novel precision nanotherapeutics or the repurposing of already approved epigenomic pharmaceuticals. And the exosomics offers a new omics paradigm to investigate the totality of non-genetic factors contributing to disease from conception onward. And so there's a lot of people to thank and acknowledge, particularly at NINDS. And with that, I will stop sharing here and look forward to some discussion.
Well, thank you very much, Setty. I really appreciated your presentation today. This is really nice to hear about this intersection of fields that's coming together. And in fact, I think one of the people who was interested in attending today had emailed me and said, "Is there a paper that I should be reading before I come to this club?" Because I have looked, I've tried to look up some of these keywords on PubMed, and I didn't find anything that included all of the keywords. So indeed, I think this is a very interesting area for you to be exploring. And again, congratulations for now starting up your independent research program. That's wonderful. So we have quite a few questions that have come in, and I want to start with a few that come from people whose microphones are not working. So Yvonne is asking, the active MS patients, she assumes are in the relapse phase. Have you considered looking at differences between relapse and remission phases, and could they be used to predict progression in any way?
Yes, so there are various approaches here in terms of studying patients during their relapse or in remission. And certainly that's something that we will look at. I think for this pilot study, it was really, can we find differences between not so much the active versus the stable patients, but perhaps the pediatric versus the adult. That's one area that I'm interested in, because there is something different about the pediatric patients who have a developing nervous system and a developing immune system, and how they present with MS versus the adults. And we don't necessarily know why. And so there are certainly groups that are looking at biomarkers to differentiate between active and stable and secondary progressive MS patients. And so that would be an area of future research. But for this particular study, it's a small cohort and it's just a pilot. But yes, that's a relevant question.
Good, and we'll see if Yvonne has any follow-up to that. But I'll go on now to a second person whose mic is not working. So in your CSF investigations, SEC fractions, what are some of the non-EV markers? So these depleted or absent markers that you might have tested during your characterization experiments?
So that is a great question, and I would love to get any suggestions on that. We haven't yet pursued that aspect of characterizing the non-EV markers, but that is something that we are in the process of doing. And if there are people who have had good success with that piece, I would love to hear your thoughts.
Yeah, we have a lot for some of the other biofluids, right? So we know, especially in blood, a lot of markers that we don't expect to find in EVs and that can indicate how pure our preps are. But what is that going to be in CSF, and can we come to a consensus on that? And I guess that's something that the task force is also working on right now. So
right. Yeah.
Good. Okay. So we have a question here. Two people actually alluded to this. So Rena Kartha, do you want to go ahead and ask your question here?
That was really nice talk and an interesting area of research. Thanks, Dr. Magna. So my question was, I thought when you were showing the background information, you talked about data from plasma, but then in your research, you were using serum. And I was wondering if there was any rationale behind it, or if you have compared serum versus plasma markers and decided to go with serum.
I'm not recalling exactly what background information, but yes, we just wanted to start with serum before moving to plasma. So it was just, not any biological reason other than the serum will be easier for us to characterize than the plasma. So that was why.
Very good. Let's go next to Zach. Zach has a very interesting question here about some remnants of our evolution.
Hi, great talk. So my question relates to the human endogenous retroviral protein syncytin 1. It's a fully functional viral envelope fusion protein that's in our DNA still. So I know it's been shown to be upregulated in placental development in the placenta, but it's also been associated with multiple sclerosis. And I was just curious if you've looked for the presence of this syncytin 1 protein on the presence of the EVs, because as a viral fusion protein, it could hypothetically get on the EV surface and potentially mediate membrane fusion with the target cell, delivery of bioactive cargo. So perhaps that could have some association with the inflammation or whatever effects you're seeing from EV-mediated cargo delivery. So I just wondered if you've had any opportunity to look for that, or if you saw that protein or anything like that.
Yeah, thank you for the question. That is a very interesting area of investigation. Actually, when I was doing my PhD, I did a meta-analysis looking at these endogenous retroviruses in MS. I haven't looked at them in terms of the EVs or the samples, but that's certainly something that Avi Nath's group at NIH does a lot of, and working with them, that's something that we briefly discussed, but didn't make a priority here. But yes, something to be mindful of.
Thank you.
Very interesting. Norm Howie, about adipose-derived EVs.
Thanks, Ken. And I'm sorry, it's a very simple question. First of all, thank you for the talk. Very interesting. I really love the idea of adipose-derived EVs playing a role in all kinds of metabolism. It's really understudied. And my question is simply, what markers are you using to isolate adipose-derived EVs? There's a few publications out there, and I'm a little skeptical at this point of how well we can isolate those. Mm-hmm. And I'm sorry if you mentioned it in your talk and I blanked out for a second and I missed it, but if you could tell us, that'd be great.
Yeah, sure. So that is, this will be the first time I try to isolate adipocyte-derived EVs, and the work that has been published, at least some of it, people have used fatty acid binding protein 4. I don't know that it's well established whether that's on the surface of EVs or cytoplasmic. I think, my approach, and I would welcome any feedback, was going to be to use the QEV to characterize the different fractions of our EVs, and then probe for some of the adipocyte specific markers. So fatty acid binding protein 4 is one of them. And some of the adipokines, so leptin, adiponectin, we actually just starting to characterize our EVs from the NIH project. We have, at least our antibody works really well for the adiponectin. We haven't found it yet in the EVs, but so those are some of the approaches I was going to take, was just to try to characterize the different fractions and see if I could find any of the adipocyte enriched markers.
Okay, thank you. Unfortunately, I can't offer any additional suggestions, but maybe others all can.
Well, since we also had a question here about adipokines specifically, so let's go ahead to, is it Fanuel or Fanuel?
Fanuel, it's fine. So my question is, I probably missed it. You said it, but I'm not sure. So did you have any evidence of the presence of the adipokine inside the EV? And if you know which one, and if there is a difference in between groups? This is my first question. And I have another one, but it's not related. So.
Okay, so just to make sure I understood it and heard it correctly, you're wondering whether we have any evidence of adipokines inside of EVs, and if there was any difference between the patient groups?
Yes.
Okay, so there are studies that have been published characterizing adipocyte-derived EVs based on some of the adipokines. The project that I presented is one that we will be starting. I haven't started it yet, so I can't comment on, at least from the EV standpoint, but there's a lot of literature out there in terms of adipokines in multiple sclerosis patients and being different in, active versus stable, being related to more severe disease, being related to therapy response. One of the challenges in the field is that they're not always reproducible across different cohorts. And so nobody has looked at adipocyte-derived EVs in MS yet.
The other question I did have was that, I don't know if you mentioned, but I remember you have 10 bariatric samples, right? Are any of those with MS or no?
So the bariatric, pediatric cohort, no, they're non-MS.
That would be great to have.
Yeah, I would love to find, obese MS patients down the line, but these were pediatric, bariatric patients, not with MS.
And I didn't write it, but if I can ask you one little small question I got when you were answering others, I'm super curious about it. Which is the adiponectin antibody that worked well by Western blot? It was very difficult to find it. I mean, for me, I was curious which one.
If you can email me, I can email you that information because I don't have it off the top of my head.
Okay, sure. Okay, thank you.
Mm-hmm.
Great, thanks. And James Drummond asks about pediatric epilepsy. Have you thought about adding any samples from pediatric epilepsy?
Yes, so I'm, within the realm of pediatric neuroimmunology, there are autoimmune epilepsies, and that is certainly, a cohort that would be interesting to study. I don't know that I would include them necessarily right now in these studies, but definitely that's one area just for studying CNS inflammation, looking at EVs in those patients. And so working with my neurosurgical colleagues who do epilepsy surgery, as well as my epileptologist colleagues, we can certainly look at that cohort.
So that's what I was thinking, is you might actually get a bigger cohort faster with epilepsy. Yep. And there is an inflammatory component to at least a lot of them.
Definitely, yes.
Thanks for the suggestion. Patrick James asks, is there a correlation between EV composition coming from the adipocytes and those in the CSF from the same patient or participant? Is that an experiment that you've been able to do?
We haven't done that. However, there are some papers out there looking at adiponectin in the CSF of MS patients, but we have not looked at, literally nobody has looked at adipocyte-derived EVs in MS yet.
Let's go next to Susan, who has several questions for us here. Susan, go ahead.
I had a couple of questions. One was, if you're going to use human-derived, if you believe epigenetics is an important element in MS risk or progression, using iPS-derived or those sorts of cells, how are you going to? Do you think that's going to work, basically? If you've got these iPS cells and actually in MS, it's epigenetic.
So for the modeling, you're referring to? Okay, so I don't know the technical aspects of the biomimetic platform. However, one of our collaborators in that group has some expertise in nuclear reprogramming of cells and using those cells. So what they've done to create a cerebrovascular unit model is reprogram fibroblasts to endothelial cells to look at the blood-brain barrier. And so I think that's something that we'll have to discuss when we get to that point, definitely.
And then the only other quick question is, or suggestion is, couldn't you use the supernatant from your fat cultures?
Yep.
To then characterize the cell surface markers that might be on adipocytes in order to have a better handle of where to start, at least.
Right. Yes. Thank you. Yep. So I plan to, so the adipocyte, so the adipose tissue is going to have, fibroblasts and macrophages and other cells in there. And so I do plan to characterize that, the supernatant there, and look at what vesicles might be there to try to better define, what are we going to say are adipocyte derived EVs? Yep.
Yeah. Exactly.
Thank you.
Thanks for the questions. And let's go next to Francis, Francis Werter.
My question was, is there any data showing crosstalk of EVs during pregnancy across the placenta? Because you had suggested maternal fetal crosstalk. Can that be measured?
So that's a good question. And that's, I haven't yet delved too deep into the placental crosstalk, other than to note that there are studies out there, mainly in animal models, but also at least in human studies, just collecting or isolating EVs from pregnant women with various pregnancy disorders. Now, there was one really fascinating article that looked at, it was obviously an animal model, but looked at the role of EVs in transmitting intergenerational stress to the progeny in the context of neurodevelopment. So I do think that is an area that is understudied at this point, but could be of potential relevance.
Thank you. Thank you for the question. Lilia Panushkina.
Thank you for the talk. It's just not a question. It's like an idea just came to my mind when you've been showing your 3D systems with the multi-layered. And so, of course, it's amazing, but I guess it's a little bit complicated to control the vesicle production over there because you got like the complex system with many different issues. And I think that's been said already before by Susan also, that the cell culture maybe is going to be a really good idea to start with, at least to evaluate the surface biomarkers for adipose derived EVs. But maybe like just instead of, with my preference in that case, going not to the 2D cell culture, but to 3D like organoids or that sort of modeling may be interesting also for you. I don't know whether you tried it or not, but that's just an idea. Maybe it could be also useful, especially in the case with that tissue, that the 3D environment is probably important in this complex talking between the immune cells and the neural. So that just was in the comment.
Yes.
Yeah, thank you for the talk. It was fascinating.
Thank you.
We also have let's see here. This is an experimental design question from Raphael Schneider.
Hi, I'm Raphael. I'm also working on multiple sclerosis. I'm a neurologist in Toronto. Very interesting talk. And we've been struggling with the question of whether or not to pool the different fractions and which fractions to pool, because there's a lot of groups that do that. And because our feeling has been that if we're starting to analyze single fractions, there's actually quite a bit of heterogeneity if you say, look in fraction 10 of different individuals. So what's your feeling about going for single isone fractions versus pooling several fractions together?
Yeah, I mean, that's a very important question. I personally think that it will depend on what your, primary outcome is, what you plan to do. And so for my initial pilot studies, once I have characterized the different fractions just to get a sense of, the heterogeneity and so forth, I would not have a problem subsequently pooling fractions just to increase, the volume and to be able to do further downstream assays. I guess it depends on whether there's a particular component within a given fraction that you're interested in, focusing on. But I think for the purposes of at least trying to evaluate inter-individual differences, I think pooling would be fine. I wouldn't pool, for example, in my studies, I wouldn't pool all my active MS and then compare it to the the stable MS, but I think within an individual, it would be fine to pool depending on what your question is.
Okay, thanks. Thank you.
Thanks, Raphael. So we have one more question and one comment. And so the question is from Modeline Longjohn, and she asks if you have looked for brain cell markers in your serum samples. So are you seeing any evidence of oligo or neuronal EVs that are there in the serum?
So we haven't personally done that yet. That is an area that our collaborators at NINDS who are still doing the work, and now I have moved on. That's an area of interest for characterizing EV repertoires, specifically with brain-specific markers. So the short answer is I have not done that yet.
All right. And then finally, Yvonne is just pointing us back to the work of Ian Sargent and Chris Gardner et al., who were using perfused human placentas, and we're looking at EVs in that system. So among the groups that have worked on placenta and possible maternal fetal crosstalk with EVs. So also a very exciting area.
Thank you.
All right, well, this brings us to the end of our EV Club for today. So Setty, thank you so much for joining us. Thanks for presenting your work. And we certainly wish you all the best and look forward to hearing more about your work as you continue in this very exciting area.